马上一起去。
我要买。
你穿的那个衣服。
Via Zoom. So these lectures are going to be recorded. They'll be broken up into 10 to 15 minute bite-sized pieces, and they will be live on the website every Monday morning for that week. So if we move in, I just wanted to go into the structure of how the lectures are organised. So we've got the lecture number, the broad topic, and then a layer language description. So today, fundamentals of biochemistry, just looking about what are those key things about biochemistry that you're going to need for future lectures. What I then also give you is a list of study aims for each lecture. And this is really useful because you can, as it says here, after completion of the lecture, you should be able to describe the classes of biological molecules and so forth. And you can see them here. There are these check boxes on the right-hand side. And what I would suggest is that once you go through it, if you're happy and you understand the content, just give it a tick. If you don't and you need help, and so imagine that this one here is not present, then this is where you would ask for help, as it says here, either in the tutorial, in the discussion board, or by email to make a time to come and see me. Please do make the time to ask for help. The content can be challenging, it can be complex within biochemistry, so it's really important that you make use of these opportunities to ask for help. As you're going through the content, you might also have additional questions that you might want to ask me. And again, there's lots of space here for you to put these questions as you're going through the lecture notes and then ask for help. And any biochemistry textbook is going to be suitable, but this is the one that the notes are taken from, to Moscow. And I've also tried to reference the page numbers and the chapters where possible. Sometimes it's not, but most of the time you will have a chapter and an allocation. Okay, so let's get started. So in terms of the biological molecules, you would already know the 4 classes. So we've got proteins, carbohydrates, lipids, and nucleic acids. And every organism, living or non-living, and this is why we say viruses here as a non-living organism, are composed of each of these 4 classes of molecules. And we're going to go into detail in each of these throughout the semester. We will start with proteins, so today's lecture and the upcoming 2 lectures on amino acids. We'll then move on to enzymes and move into carbohydrates, the metabolism of carbohydrates. We'll talk about lipids, and we'll also then talk about nucleic acids. So we'll talk about DNA, RNA, control mechanisms, etc. So we will touch on all of these as we go throughout the semester. So when we come to talk about proteins, hopefully you already know that these are composed of 20 amino acids. And it's the sequence of the 20 amino acids within a protein, so obviously going to be more than 20 within a protein, but the combination of those 20 that form a particular protein. And so these amino acids are linked together by what is called a peptide bond. And I know that in chemistry, you may also hear this referred to as an amide bond because of the amino group that is used, but we're talking about the same thing, and I would typically talk about it as a peptide bond. Now, when you join these together, and it can be either a very short sequence or a very, very long sequence of amino acids, these come together and they form a polymer. So remember that poly means many. And so a polymer is just many amino acids. And this is the abbreviation, amino acids, that are joined together. And once this polymer is made, they will fold together to form a 3-dimensional structure. This 3-dimensional structure is important for the function and the activity of the protein, and as it says here, key for biochemical reactions to proceed. So therefore, if we change this structure, either through a mutation or through a drug or some sort of inhibitor, we're going to change the ability for that biochemical reaction to take place. It's a very simple concept, but a very powerful concept because it really forms the basis of drug treatments and how we might interfere with, say, pathological conditions. So one other thing that you'll notice in my lecture notes is that I try to give you as many diagrams as possible, and you'll also notice that there's lots of empty space around the lecture notes. So this is where you can annotate and put extra information. So what you can see here are the 20 amino acids, but we know that there are 20, and these are going to be all unique amino acids. They come together and they are joined individually, and this is a repeat type bond that you'll see in the next lecture. So we've got one here, and so on. Okay, joining all of those amino acids, and this is the amino acid sequence. So the combination gives rise to that protein. And then these will come together to give us a three-dimensional shape, which is what we see here. And the shapes that we see here, this is actually called an alpha helix. We will talk about more in the next lecture, but we'll go into more detail about this particular shape. But you can see that this shape is quite a important shape for this particular protein. It has a particular structure, and as I mentioned just here, that's important for those biochemical reactions to take place. So make sure you understand this basic idea, but as I said, the next lecture will go into it in more detail. Okay, the next grouping, and everyone will have heard of carbohydrates. So this is a class of biological molecules that are used as a fuel source for most organisms, and of course we are no different. We use carbohydrates as a source of energy. And the most common carbohydrate that we're going to be talking about and that is used by organisms is glucose, and you can see the diagram that is down below. Now, we use glucose for energy, but when we have excess glucose, what happens to it? It's actually stored within the body as glycogen, and this is for animals. So you'll see glycogen is an animal-based store of glucose, and in plants, it's stored as starch. So for example, if we think about potatoes, we know that they are high in carbohydrates, but they are also high in starch. And so remember, we said that glucose is a carbohydrate, and this is the form of storage. This is a very classic structure of glucose. So the six-membered ring, you've got the hydroxyl groups, and these groups are going to be important for the bonding to occur. Now, as will become apparent later in this lecture, we are talking about biochemistry. You do need to get familiar, you do need to get comfortable with the chemistry aspect, because we are going to be talking about the chemical structures. We're going to be talking about those regions where a bond occurs, as we saw just here with a peptide bond. So we're going to be talking about an amine group and a carboxyl group in this context. And down below, we're talking about different type of linkages that can link glucose. So this is all information that is coming together. So make sure that you get familiar with this chemistry. Now, sometimes that can scare students, but the chemistry, or the focus of the chemistry, is going to be generally much bigger picture in terms of what are the general chemical reactions. You will not be required to know every single chemical name or structure. The ones that are important will be highlighted to you, but generally if you have an understanding of the basic principle and the control point of the chemical reaction, that is going to be sufficient. But remember, if in doubt, just ask for help. More than happy to help you. Okay, next one are lipids. So these are commonly called fats. Okay, every person will call something like this a fat. And we know that a fat or a lipid has two regions. It has a hydrophobic region, which refers to water repelling. So hydrophobic, it's afraid of water, and a hydrophilic, or a water-loving region. And so what you would have learned in first year is that this allows for the formation of the cell membrane in mammalian tissues or eukaryotic cells. And so this allows us to have an inside of a cell and the outside of a cell. And by doing so, it actually allows for us to have a cell, to have a unit of life that can exist. So very, very important molecules. But apart from this, these are also very important energy stores. So we saw above that carbohydrates are used as energy, and that's fine, but you are limited in terms of the amount of storage of carbohydrate that you can have. And similarly, we are limited by the amount of energy that can be generated through the metabolism of carbohydrates. And so fats or lipids are a much more efficient store of energy, and we will see this when we come to talk about the lipids lecture much later in the semester, and the process called beta oxidation, which is how we take fats and convert it into energy. And remember, when we're talking about energy, we're talking about the molecule ATP, adenosine triphosphate. You'll hear about this lots and lots in biochemistry because it is one of those key molecules. Now here, down the bottom, we've got a diagram. So you can see this is a space-filling model. So these spheres are meant to represent carbon, hydrogen, and oxygen atoms. And you can see that you've got this hydrophobic region and you've got this hydrophilic region, so quite distinct areas. A simplification of that would be this shorthand depiction. So you can see the two long tails that we saw on the left and this hydrophilic head on the right. And one way to keep this happy, so remember, if we've got an aqueous environment on the outside, these hydrophilic regions are going to be happy, and these hydrophobic inner regions are going to be protected from the water. And so this allows us to have an internal and external portion of the cell, which is really quite critical. Now, lastly, we have nucleic acids. And you would know that nucleic acids are the way that we both store and transfer genetic information of an organism. And so we've got RNA or DNA, depending on the organism. Most of the time we're going to be talking about DNA, but we know that viruses, for example, will use RNA. The differences are quite minor in terms of the structure. There are some inherent differences in terms of the stability of the molecule, but we'll go through those basics in later lectures near the end of the semester. Now, when we look at nucleic acids, they're composed of just 4 nucleotides. So we've got adenine, or A is the shorthand, cytosine, C, guanine for G, and thymine for T. And here we are just talking about the DNA basis, because what we'll see is that with RNA, this is actually uracil instead of thymine. So there's a difference in the single base. We've covered that there are the two main types. There's ribonucleic acid and there is deoxyribonucleic acid. And this is a structure of the double helix. And what you can see here is that you've got a yellow and a purple. The yellow might be the base A and the purple might be the base T. If you look at this diagram, you can see it's always yellow and purple, yellow and purple. These are constrained in terms of their bonding. The other where you've got orange and green might be cytosine and guanine. And again, here's a constraint. G to C or C to G will always take place. And this is how the DNA is structured. So this information then allows us to come into what is referred to as the central dogma of biochemistry. And we'll talk about this over and over throughout the semester. If we talk about a eukaryotic organism, the DNA is involved in the replication of the cell. And as a result of all of this occurs in all of the cells, this is going to occur in the host organism. The DNA is transcribed through a process called transcription into RNA. And so this is basically reading the DNA and converting the message to RNA. Okay, that's what transcription is. So if you want to transcribe a document from one language to another, you copy it, you might go from English to Italian, for example. That is transcription. It's the same thing here. We're going from one type of message, DNA, to another, which is RNA. The RNA, in turn, acts as a messenger molecule. And it is a messenger typically when it is read or translated into a protein. And the proteins, what do? The proteins are those biochemical, allow for those biochemical reactions to occur within cells, typically as enzymes. Okay? So we will go through all of this. We will talk about proteins, we will talk about RNA, and we will talk about DNA and this central dogma. So this is what the diagram is showing you here. We have DNA. DNA is replicated to renew the cell, and then sometimes the DNA is copied or transcribed into RNA, and that RNA is then translated into a protein, and the protein then does some sort of work, some sort of enzymatic process within the cell. Okay, I'm going to stop there, because as I said, I want to keep this short and sharp, but when I come back, you can see at the bottom, we'll start to talk about some key aspects. Okay, welcome back. So this is part 2 of lecture 1. So we finished on the previous lecture talking about the central dogma, and now I want to talk to you about the key aspects of cells and organelles. So as you already know, the cell is the basic unit of life, and so we either have an individual cell or we have a multicellular organism, and we will talk about prokaryotic cells or eukaryotic cells. And so obviously we fit within the eukaryotic category. Prokaryotes are things such as bacteria. Now when we look at the key organelles within a eukaryote, you can see that we've got the cross-section or an image of a eukaryotic cell. We can see the nucleus where the DNA is held, the Golgi complex, we've got lysosomes, we've got mitochondria for energy, and we've got the endoplasmic reticulum. And again, you can see the same thing in this diagram shown a bit more graphically to show you those key organelles. Why do you need to know about these organelles? Because distinct activities take place within these organelles. They're important for the basic biochemistry of the cell, and in turn they're going to be important for the basic biochemistry of the organism. So let's move on. So if we start off, we have obviously the plasma membrane. And so as we saw when we were talking about lipids, lipids have hydrophobic and hydrophilic regions, and because of that, it allows for the creation of a membrane that separates the inside from the outside environment. And this is really central to the formation of a cell. Eukaryotic cells have membrane-enclosed organelles. So the organelles are encased within their own individual membrane within the cell, whereas prokaryotic cells do not. That's one of the key distinguishing features. And as I said, we will talk about both of these. And when we do, we will usually start with a prokaryotic cell, get the fundamentals under our belt, and then we will move to the eukaryotic cell, because the process, if it's a transcription or translation, is simpler within a prokaryotic cell, and it is similar within the eukaryotic cell, but just with a little bit more complexity. Now, we've spoken about the inside and the outside, and previously I spoke about the aqueous environment or the water environment, and this is referred to as the cytoplasm. So this is the inner fluid region of the cell, and this is where lots of those biochemical reactions are actually going to take place within the cytoplasm. Not all, but most of them. But what you also have is what is called the cytoskeleton. So just as you have a skeleton that gives you your shape, a cell has a particular shape. So there are proteins within the cell called the cytoskeleton that allow it to conform to a particular shape and also to change shape in response to an external signal. Now, we spoke briefly above about the nucleus. And so again, this is a double membrane organelle, because remember, we are looking at a eukaryotic cell, and this is where DNA is stored in these cells. Why do we have this double membrane? The double membrane allows for the formation of pores. So these are openings that allow for things to enter and exit the nucleus. And why would that be necessary? Because this is where our DNA is stored. This is where the DNA is stored. We need to have access to the DNA to either to copy it, to transcribe it, or to replicate the entire genome. And so you can see here, very close, if you imagine just looking at this portion up top, okay, you can see that there's one line and another line. That is that double membrane. You can see the endoplasmic reticulum on the outside, and this is the nucleic acid on the inside. Now we'll get to the endoplasmic reticulum shortly. You can see endoplasmic reticulum on the outside, and this is the nucleic acid on the inside. Now we'll get to the endoplasmic reticulum shortly. The next major organelle that you should have heard of is the mitochondria, and this is the plural mitochondrion for one. This is the organelle where energy is generated. And remember, when we are talking about energy in the context of biochemistry, we are talking about the production of this molecule, adenosine triphosphate. Okay, so whenever I talk to you about energy within the cell, it will be around ATP for 99% of the case. There are one or two situations where we talk about other molecules called GTP, or another molecule called GTP, but we are predominantly focusing on ATP, and certainly within the mitochondria, this is where we talk about this, because this is where it's made, and this is why the mitochondria is so important. And again, it too has two membranes, and the benefit of this is that it actually increases the surface area within the mitochondria. So there's a diagram down here, you can see these double layers. And what that does is that by increasing the surface area, it gives more area for reactions to occur. And remember that the reactions are here to make ATP. So you can see all of this invagination that we've got on the surface, and in between those gaps are where the reactions are going to take place. So very, very important. We will come to talk about this in the lecture on oxidative phosphorylation. So this is basically how do we make ATP? How does the membrane or the mitochondria make ATP? So obviously very, very important topic. The next one I want to talk about is called the endoplasmic reticulum, or short for ER. And we've got smooth and rough ER. The smooth ER doesn't have anything attached to it, and so it just looks, by its name, smooth. And this is where chemicals, exogenous chemicals such as drugs, can interact and be processed. The rough endoplasmic reticulum, which is really the main one that we're going to be talking about, it is rough because it is studded with ribosomes. And the ribosome is the organelle that is involved in protein synthesis. So this is the organelle that reads the RNA, and more specifically messenger RNA, and converts it into a protein. So this is that translation event. So if you look at this diagram here, you can see here is the cytoplasm. This is a cross-section of the endoplasmic reticulum. You can see it's closely associated with the nucleus at the top. You've got some regions here that are smooth, so you can see where I'm just shading in. There are no ribosomes. And then you've got these other areas that are studded with lots and lots of ribosomes. And notice, the ribosome specifically RNA and converts it into a protein. So this is that translation event. So if you look at this diagram here, you can see here is the cytoplasm. This is a cross-section of the endoplasmic reticulum. You can see it's closely associated with the nucleus at the top. You've got some regions here that are smooth, so you can see where I'm just shading in. There are no ribosomes. And then you've got these other areas that are studded with lots and lots of ribosomes. And notice, the ribosomes are close to the nucleus because remember the DNA is going to get converted into RNA, and more specifically messenger RNA. It then goes to the ribosomes, and that in turn makes proteins. So you want them to be close together because they're all going to be involved in similar functions. Now, once the protein is made, it goes to another organelle called the Golgi body. And these are vesicles from the rough endoplasmic reticulum that move or come from this and move to the Golgi complex. And this is an organelle where we've got further processing. So what you need to appreciate here is that a protein that is freshly made, okay, it has two options. It can be used, or it might need to be modified and then used. Okay, so just as you would imagine, if you make something, sometimes you can make a protein and it might be functional immediately. Other times you have to modify it because this enhances the activity. So for example, you might have to add a carbohydrate to that particular protein. You might have to add a lipid to the protein. And if you add a lipid to a protein, that can actually force it to go to the cell membrane and help it to target and traffic within the cell. So really important in terms of that location process. And the last one I think that I want to talk about is called the secretory granule. And these are buddings or bits that pinch off from the Golgi complex. And what you have here is that they will pinch off and travel to the cell membrane and then release their contents to the outside of the cell. And remember, the outside of the cell is going to be it might be into the bloodstream or it might be into the lymph. So this is an important mechanism. So here you see all the organelles we've seen. We've got the nucleus, got the endoplasmic reticulum. You can see in this diagram it's shown as the rough endoplasmic reticulum with all of the ribosomes. You've got that pinching off going to the Golgi complex, and this is where that processing is occurring. Once that's occurred, these pinch off and migrate, so they keep going this way, migrate to the surface of the cell, and whatever was inside, these red dots, which are likely to be proteins, these are then released as a secreted protein. And then these mediate and effect. So they do something to the organism. They might cause it to grow, for example. So this could be a growth hormone release. So there's lots of different things that can occur. Okay, I've got a few more. The other ones that we want to talk about are the endosome, and this is slightly different. So here we've got, it's involved in taking material from the outside of the cell and bringing it to the interior. So this is the opposite of what we just covered. Previously we saw the lysosomes releasing, the endosome is bringing it in. And this is called endocytosis. And we do this, or the cells do this, because they want to transport iron or vitamin B12 or cholesterol into the cell. Now, you would have heard of iron because this is important in oxygen uptake. Vitamin B12 is important for red blood cell formation, and cholesterol is actually important for the cell membrane. So this is that way of bringing things in. And the last one I want to talk about is the lysosome. And this is an organelle that has digestive enzymes. So you can think of it as a mini stomach of the cell. So when things are internalized, as we've got here with the endosome, the enzymes that are there are used to degrade that ingested material. And in most cases, this might be some organisms like a bacteria that have been ingested, and this is a way of killing them and removing them from the host. So it's a very simple way of clearance just by ingestion and digesting them. So here is a diagram. So here we can see an organism or a cell. You've got these arrows. Those arrows are bacteria that have been ingested, and we've actually got a white blood cell, so a macrophage, and now those endosomes or lysosomes are going to migrate towards those bacteria. They're going to be digested, and they're going to be killed off. Okay, I'll stop here for this part. We'll move on to some basic biochemical reactions in the next recording. Okay, welcome back. So this is part 3. Just move a bit further down. Now, as I mentioned before, obviously this is biochemistry. We're going to have to talk about biochemical reactions. Now, all of the biochemical reactions that we have within a cell take place within an aqueous or a water environment. And so when we look at a cell, 70% is going to be water. But you might ask the question, well, why is water so important? Now, when we look at water, we know that water is H2O, so 2 hydrogens, 1 oxygen atom. Oxygen in water is electronegative. And what does that mean? It means that the electrons in the hydrogen, so remember we've got the 2, draw it like this, are attracted to the oxygen atom. So the electrons want to move in this direction. The oxygen is more electronegative because remember, these are negatively charged electrons, they're going here, so therefore the oxygen becomes more electronegative. So oxygen is slightly negative while the hydrogen becomes slightly positive. And what is shown here is the delta symbol is just to represent that this becomes negative and these become positive. You might be thinking, well, that's very interesting, but why is that important? This leads to an unequal charge distribution. And what occurs here is that water is now polar. So you've got more charge in one location, less charge in other locations. Polarity, or this polar nature, is important because it allows for the formation of bonds called hydrogen bonds. And hydrogen bonds are very common, or ubiquitous, with water. Now, these hydrogen bonds are important because they're at the core of a chemical reaction within a cell. And for a chemical reaction to occur, it needs to have some characteristics. The first one is that the reactions need to be weak because you want to be able to make and break bonds readily. They need to be reversible, so if you have a forward reaction, you want to be able to go backwards. And they also need to be non-covalent because, again, that facilitates the formation and the breakdown of these bonds. And so when we come to talk about these non-covalent bonds, there are 3 fundamental types. There is ionic bonds, which is basically electrostatic charges. There are the hydrogen bonds, and there are van der Waals bonds. So let's just go into those in a little bit more detail. Ionic bonds. These are interactions between distinct electrical charges on an atom. So if you've got a negative component, it's going to want to be attracted to the positive. If you have another negative component on an atom, you're going to end up with repulsion because the same charge will want to repel each other. So this is a very simple way of interacting. What we know is that water will also weaken electrostatic interactions because you have got negative regions and positive regions. Water is a ubiquitous molecule, so it's able to interact and break those bonds apart. Remember, we want things that are weak, reversible, and non-covalent, so water is going to play a major role there. We've got hydrogen bonds. The 2 most electronegative, the ones that have the most negativity in terms of capturing the electrons, are going to be oxygen, as we saw, but also nitrogen.
Now, hydrogen bonds on their own are actually very, very weak, which remember we set up here is one of those characteristics. A weak bond is something that can be formed and broken very, very easily. And that is important because if it's too strong, the cell is going to have to either put in energy in the form of ATP, or it may not be able to break that bond. And that then becomes problematic because things terminate. So here's an example of a bond between a donor and the acceptor. So here we've got a hydrogen atom. So you can see nitrogen is electronegative. The hydrogen has donated its electrons. And because we've got this charge-charge interaction, we've got a hydrogen bond occurring. Same thing here with the oxygen atom, and then you've got the different combinations, oxygen and hydrogen, and again, just changing the order. It's showing you the same thing, just with all the different possible combinations for either oxygen or nitrogen. So it's just giving you an idea. Now, that will cover the vast majority of bonds. But what about molecules that are not polar or charged? What do we do there? This is where the van der Waals bonding comes into play. Because remember, we had ionic for the charged bond, we had hydrogen, and now we've got our third grouping, which is the van der Waals. So as with hydrogen bonds, these are individually very weak, but they can be strengthened when you've got multiple bonds existing. So one hydrogen bond or one van der Waals is weak. But if you've got 100 of these, then it's going to be stronger. So the more interactions, the stronger the bond, but individually they are still weak, which means that individually they can be broken quite readily. Okay, just to finish off this lecture, we've got some common chemical functional groups. So we've spoken about the functional group being hydrophobic. So aliphatic, these are carbon-hydrogen chains. You can see that here where we've got a methyl group, and here we've got an example of an amino acid. You can also see the same thing with an aromatic hydrocarbon, as we've got here. Remember, aromatic is just referring to this group, the six-membered ring. And again, here we've got another amino acid, phenylalanine. We've also got the key groups. We've got the hydroxyl group, which is called an alcohol, and this is the group here. So we've got ethanol, and you've got aldehydes. This is the grouping here, and you can see the C double bond here, so acetaldehyde. We've got ketones, same thing, but the R groups are different on either side compared to the aldehyde. You see that here, so we've got acetone, and we've got our carboxylic acid, which is a COO, which is what we can see here. And you see these are shown in different directions, but this is acetic acid, and acetic acid is what you have in salt and vinegar chips, for example. If we move further down, we've got the amino groups, or amines. This is the NH2 group, which I mentioned previously, and here it is. This is part of an amino acid, so alanine, and you've got phosphate groups, organic phosphate. So remember, adenosine triphosphate, ATP, it's got 3 of these. This is the phosphate group, and here we've got a molecule with a phosphate group attached. And we've also got sulfur hydryl group. So a sulfur hydryl group is telling you that it has a sulfur atom. These are called a thiol group. And you can see that in cysteine, which is another amino acid, we have this sulfur hydryl group here. This is important because the sulfur hydryl can also go to a disulfide bond, which is we lose the proton, and we just have 2 sulfur groups coming together. Now, why are we talking about these chemical reactions? Because we will see each and every one of these, all of these groupings in all subsequent lectures to some degree, to some extent. So when I talk to you about a carboxylic acid and an amine group in the next lecture, you'll know what these groups are. You'll have an appreciation of the bonding and how they come together to form a peptide bond or an amide bond. So it's very, very important. Okay, before I finish, just to remind you, at the end of every lecture, what I've tried to do is give you a short chapter. So, the other thing that I didn't do in the first lecture, but I do subsequently, is that I like to give you a bit of an introduction at the beginning. So usually the first paragraph is going to summarize all of the key points that are going to be coming. So use that as a guide so you know, okay, what the next section is going to be, what's the same sort of content or the likelihood of the content as it's coming. So, what are the key properties of proteins? We spoke in the first lecture, proteins are made up of individual amino acids, AAs, and so proteins are polymers of amino acids. And the order of the amino acids in a protein is what differentiates different proteins. So if we change the sequence of amino acids from one protein to another, we have now generated a different protein. The function of a protein is very, very much dependent or highly dependent on its 3-dimensional structure. So again, if we change the 3D structure, we can affect its activity. And if we change the sequence, we change the 3D structure. So the two are very much interconnected. Amino acids have got a wide range of functional groups. So think back to the last portion of the last lecture where we spoke about alcohols, thiols, carboxylic acids, and basic groups. We will be talking about those a lot. And the functional groups, these functional groups affect the activity of individual amino acids and therefore the overall protein. So if you have a mixture of basic proteins or basic amino acids in a protein or more basic amino acids in a protein than other groups, then the protein in general is going to have a basic nature. And by basic, I refer to pH, not complexity. And lastly, proteins can interact to form very complicated assemblies and have complicated functions. And one of the best examples is DNA replication, where we've got a whole myriad of individual proteins or complexes of proteins coming together to perform a really complicated function, such as copying DNA. Okay, so proteins are made from amino acids. We've covered that. So all amino acids have a central alpha carbon atom. If we go to this diagram, it's referring to this carbon atom here. This is our alpha carbon. The alpha carbon is bonded to an amino group. No, amino group, nitrogen's missing, so it's there. Make sure you update your diagram. I will update it as well. There's a carboxylic acid group, which is here. And there's a hydrogen atom at another location. So all amino acids have this portion in common. But what we have is also what is referred to as an R group. And this is where we have, or they will vary at this particular location. So each amino acid has a unique R group, and this can also be referred to as its side chain. Now we have 20 amino acids that exist in nature, and these 20 are used in various combinations and numbers to make individual proteins. Now what we also know is that most amino acids will exist in a mirror image form, so they are chiral. So you would have learned in chemistry that there is the L and the D isomers. What you need to remember, this is important, only the L version are found in proteins, so we don't use the D version in our body. And so you can see here is the L isomer, amino group is on this side, and obviously the mirror version is for it to be on this side. But also we've got the conformation of the other molecules in this particular group. So this one here, we don't really need to worry too much about, but I bring it because you do need to understand the difference between the two groups. Now, I've also tried to give enough detail in the legend. So what you can see, basic structure of amino acid shown using a Fisher projection, which is this one here. Okay, this is a Fisher projection like a star. The right-hand side, which is here, obviously, shows the amino acid in both the left and the right, or the L and the D isomeric form. The amine group is shown protonated, so remember, the amine group is NH2. If you want to add an extra proton, that's a positive charge, so it becomes NH3+. And the carboxyl group, which is typically shown like this, is deprotonated, so it is shown like this, it has lost its proton. Where could that have that proton could have gone? Well, it's gone here. Okay, so we're shifting between the two. You can see that here and here in this diagram. And you would see this at a physiological pH. Now, physiological is not 7, it's about 7.4, but we're close enough to see that this is going to be the standard configuration of things. Now, physiological is not 7, it's about 7.4, but we're close enough to see that this is going to be the standard configuration of things. So following on from that, what is the effect of pH on the charge of an amino acid? Amino acids at a pH of 7 exist as dipolar ions, which are called a Zwitter ion. Now remember, dipolar or polarity was where you've got a charge distribution or differences in charge, and we just saw that the amino group was positive and the carboxyl group was negative. So here are the two dipolar regions. So amino group is protonated, carboxyl group is deprotonated. The balance between this protonated state and deprotonated state, which is also called the ionization state, because remember if they're ionized, they're going to be in these configurations. This will vary with the pH of the solution. And what do I mean by that? Well, these amino acids have to be dissolved in something in an aqueous environment. Now, depending on what the pH of that environment is, it's going to impact the amino group or the carboxyl group differently. So, if we start off with an acidic solution, so really acidic, a pH of 1, okay, so almost as far as you can go. Now, the amino group is acting as a weak base. And remember, a base will accept a proton, and acid will donate a proton. So the amino group is able to accept the protons. And we saw this because we had NH2 going to NH3+. It gained or accepted that extra proton, hence it's acting as a base. So it exists in this NH3+ configuration. We also know that if this is a base, the carboxyl group, by donating a proton, as we saw up here, must be an acid, a very weak acid. Now, if this is a weak acid and it's in a very strong acidic pH of 1, it will not want to dissociate because it is a weaker acid in comparison to the environment. And so it will stay in the COOH configuration. So in an acidic pH, there's going to be lots of protons present. So even though the carboxyl group is a weak acid, there are more protons available in the solution and it does not dissociate. So that's the example and the explanation as to why it occurs. Now, what happens as the solution becomes more basic? So as we start to move from 1 towards 14. As we do that, there are going to be fewer free protons because remember, protons are lost, we're going to have more hydroxyl groups. So the carboxyl group can now start to donate its proton. And if the solution becomes very basic, and we're saying just a pH of 9, so 2 logs above neutral, then even the amino group can start to donate its proton. So it goes back to the NH2 version. Now, these are extremes, and we don't really see them. We don't see basic environments, but we do see acidic environments within the individual. But at a physiological pH, amino acids are going to exist in the dipolar form. Okay, in this configuration. The amino group will be protonated, carboxyl group will be deprotonated. Now, highlight this piece of information here because it's really important to understand. This discussion is only referring to the amine and the carboxyl groups attached to that central alpha carbon. We haven't spoken about the R group yet, but this is true about those regions. Deprotonated. Now, highlight this piece of information here because it's really important to understand. This discussion is only referring to the amine and the carboxyl groups attached to that central alpha carbon. We haven't spoken about the R group yet, but this is true about those regions. If the R group also contains an amine or a carboxyl group, it will also be affected by the surrounding pH of the solution. So if we've got an R group which is an amine group, it can accept a proton. If we have an R group which is a carboxyl group, it can donate a proton. So we've got the same logic, it's just that there might be more or less of those particular groups present. Okay, here's a diagram which is showing you that basic idea. So at the top, we've got, let's say pH 7. Here is our central alpha carbon, our amino group, carboxyl group, and our proton. And we've got our R group. And remember, we're not showing you what the R group is here because we're not worried about that. We're just worried about the core component. And you can see the amino group is protonated, carboxyl group is deprotonated. As we get down to pH of 1, okay, we're going this way, you see that we're going to add a proton. This is already protonated, the carboxyl group can become fully protonated, and this is the configuration. And similarly, if we go in the opposite direction, we're going to a basic environment. The carboxyl group will lose its proton because it's acting as an acid, and even the proton that was on the amino group is going to be lost. But these are in extremes, so notice we're looking at 1 and 12. Generally, we're going to be in this environment here, pH of 7, so we're going to have this diprotonated form that is going to be present within the cell. And this is just a diagram that is showing you that transition state then occurs. But when you've got this dipolar form, it's this zwitterionic form. Okay, I'll stop there because we're going to get into more detail about the individual amino acids, but I'll see you in the next recording. Okay, welcome back. So we just finished talking about the zwitterionic state. Let's now move on to the R group of each amino acid. And each amino acid has a unique R group, and they will vary in their size, their shape, charge, their ability to bond via hydrogen bonds, their hydrophobic character, so whether they like to be in an aqueous environment or not, and their chemical reactivity. Are they chemically reactive or are they more inert? And so these very properties are due to the different functional groups in the R group. So you might have an alcohol, you might have a thiol, carboxyl group, an amine group, etc. You might even just have a carbon atom or a proton. So you can see that all of these are going to contribute to that, and we'll see the structures in a moment. Now, in terms of the nomenclature, all amino acids have a unique name, but they also have a 1-letter and a 3-letter code. And remember, one of the learning objectives was for you to become familiar with these words and these codes. So the abbreviations are typically the first 3 letters of the name. Now, when you come to study this, what I would suggest is that you remember these as the exceptions, okay? Because once you understand these exceptions, all the others are going to follow this rule. It's going to be the first 3 letters of the name and usually the first letter, except for here. Now, why do we have exceptions? Because there are other amino acids that start with A, G, I, and T. And so we can't use that same approach because then we would be doubling up. So symbols are usually the first letter of the amino acid, so G is going to be for glycine. This is a really nice table because it summarizes, first of all, the full name of the amino acid. So you can see alanine is ALA and arginine. Okay, A for alanine, sorry. Then we've got arginine, so it's going to be the first 3 letters, ARG, and the second letter, R. So that helps you to remember. Now, the next one was asparagine, so ASN. It's not P, because P is used elsewhere, so you can see here for proline, so this is N. So this is why you need to remember these differences, and the remainder you can remember much more readily. Rather than trying to remember all 20, you only really need to remember the first 4, and the rest you'll be able to work out. But it is important because you'll see these letterings being used, but more so, you'll see the 1-letter abbreviation, because even textbooks want to save space, and they'll use these single letter codes. So you must be able to convert between all 3 forms as we're going through the semester. So as it says here, become familiar with these abbreviations. So you must be able to three forms as we're going through the semester. So as it says here, become familiar with these abbreviations. Okay, what about the structure of the R groups? And so I've given you diagrams of all of the R groups. And you can see we've got non-polar, so those that are not polar, so there's no charge difference. Aliphatic are just those that have a carbon and hydrogen bond. Aromatic have that benzene group. Hydroxyl have a hydroxyl group. You've got the polar, which is going to be opposite to the non-polar. The basic, the acidic, and those with a cyclic or a circular R group. So let's go through them. Now, let me just remind you, this is the core of every amino acid. Central alpha, amino group, carboxyl group, proton. The only difference with this diagram to the one that I showed you before was that the hydrogen atom is at the top and the R group is at the bottom. So the R group is shown in the red box. Okay? So you see glycine is the simplest non-polar amino acid, is just a proton. It's going to be very small and not overly chemically reactive. The next one is alanine, because again, same thing, but it's a methyl group as opposed to a proton. So structure is very similar. Remember that aliphatic refers to a hydrocarbon chain. So these are just going to be carbon and hydrogen extensions, and they're just going to be slightly bigger. So here we've got valine, so carbon with methyl groups and a proton. Here is leucine, same thing, but just slightly bigger. Isoleucine, again, it's an isomer of leucine. That's what the iso is referring to, the same molecule, but just in a different configuration. And then methionine, which is a linear grouping. But I'll just bring your attention here. We've got a sulfur atom, okay, part of the R group. This is not a sulfur-hydryl group, we'll see this in cysteine, but notice that you've got these molecules, okay, unique or different types of elements that you will typically not see. The next ones you can see, these are going to be aromatic, and aromatic means that they have a benzene ring molecule. You can see phenylalanine, so we've got alanine coming up or above. Here's the group. Tyrosine also has the benzene group, and tryptophan has two benzene groups, so 6 and a 5-membered ring in this particular location. So you can see it's starting to get bigger and bigger and more complex. The hydroxyl containing amino acids, so serine as an example, has one hydroxyl group. Threonine also has one hydroxyl. Alanine coming up or above. Here's the aromatic sulfur atom, okay, part of the R group. This is not a sulfur-hydryl group, we'll see this in cysteine, but notice that you've got these molecules containing amino acids, so serine as an example, has one hydroxyl group. Threonine also has one hydroxyl group, but it has a different configuration of the aliphatic component. The next one then are polar amino acids. So remember we said oxygen and nitrogen are very good at being electronegative. So you're going to have electronegativity here, electronegativity here. So the oxygen is going to take electrons from the carbon, as you see with the double bond, and the proton is going to donate its electrons to the nitrogen here. And we see the same configuration for asparagine and glutamine. Same thing, just this one's got an extra CH2 group compared to this one. We've also got basic amino acids, and basic in this case does not mean a hydroxyl group. It refers to the amine group. Okay, so make sure you understand this is not a hydroxyl group, it is an extra amine. So aliphatic with an additional amine group here. That's lysine. Arginine, same thing. We've got two amine groups. And histidine, the amine group is in this funny sort of 5-ring structure, but again, you can see NH configuration. So it's like an amine group in this configuration here. The opposite of that is going to be an acidic group. And so you can see, remember an acid, we're talking about a carboxyl. So here is one carboxyl group. Here's a same thing, but the only difference is that we've got an extra CH2 group. So aspartate and glutamate, you can see, are going to be related to each other. And the last one is a cyclic amino acid. And this is the only one that exists in this configuration, and this is proline. You see that the R group, they bond together to form this circular or cyclic structure. So, by going through this diagram, the structure or the type of group of R group, don't worry about that. You're not going to need to know that. That information can be readily found. The name, the 1 and the 3-letter code, though, you do need to know, you do need to become familiar with, because it's part of our language, it's part of the language used in a biochemistry lab and a biochemistry course. Okay, let's stop there, then we'll move on to some of the characteristics of amino acids. Okay, well, so we finished on the previous recording, we finished on the structures of the R groups. And we could see that there were quite different types of R groups. So now I want to talk about some of the differences in the physical characteristics of amino acids, and in particular, their ability to absorb ultraviolet light. Now, why is this important? Measuring the level of protein in a blood sample is a very standard approach in working out someone's health status, so someone's sick or unwell. Now, there are lots of different methods that will allow you to quantify the protein, but most of them are reactions, so they will destroy the protein in the process. But what about methods that don't require additional reactions or simpler and easier to perform? Well, we know that several amino acids have aromatic R groups. So tryptophan, tyrosine, and phenylalanine all have an aromatic R group. Aromatic R groups will absorb light at 280 nanometers. And so what you would expect is that these amino acids are likely to be evenly distributed throughout all proteins. It would be unlikely that you would have a protein that would be missing one, let alone all three of these amino acids. Possible, but unlikely. So therefore, the absorbance of a sample at 280 nanometers can be used as an approximation. It's not exact, but an approximation of protein content without destroying the protein. And you'll actually do this both online and face-to-face in your second practical. So if you look here, we've got the extinction coefficient versus wavelength. If we scan tyrosine or tryptophan, you can see that the peak absorbance for each of these round is 280 nanometer wavelength. So if these are present, then you're going to be able to quantify the protein. We can use it to estimate the amount of protein in a sample. Okay, let's move on now to linking proteins or linking amino acids together. We need to talk about the 4 levels of protein organization, and the first one is called the primary structure, and it can be written in this way, 1 with a 0, a superscript 0. Now, what is the primary structure? It is the sequence of amino acids that join to form a protein. This is what is referred to as the primary structure. It's just the order, so it might be methionine, proline, serine, and so on, okay? This is your primary structure. Amino acids join together by linking the alpha carboxyl group of one amino acid to the alpha amino group of the second amino acid, and this forms the peptide or amide bond. We lose a molecule of water, and we have to put energy into the system. And remember, as I mentioned in the previous lecture, energy comes in the form of ATP in most cases. So here is an amino acid. So if I draw it this way, it's not as clean, but you can see the general idea. Here's the central alpha amino group, carboxyl group, and proton. And remember, with a peptide bond, we actually are not interested in the R group, so we don't need to show what the R group is because it doesn't participate. Here is the second amino acid, so this is number 1, this is number 2. You see that the amino group is close to the carboxyl group here. These come together, and notice the arrow, the forward reaction arrow is smaller than the reverse, which means that you have to put energy to force it to occur. And so what we now see is that this carboxyl group is here, this NH2 group, or the NH group that was here, is there. Remember, we've got NH3, so there's 2 protons, and we've lost this oxygen. 2 protons and an oxygen is a water molecule. That's where the water molecule comes from. It's lost through that addition. And this is now a peptide bond. And if you look at this molecule, we've now got a free carboxyl group. So if we had another NH3 group plus the remainder, you could form another peptide bond. So now that you understand how one peptide bond occurs, you understand how all peptide bonds occur, because they're all exactly the same. So it's very simple in its structure. Now, peptide bonds are actually very stable, so it's very good for the cell. And what we also know is that the polypeptide chain has polarity. So what does this mean? It has a direction. So we have a free amino group on the left, a free carboxyl group on the right. So if we look here, here's our free amino group on the left, here's our free carboxyl group on the right-hand side. And so we read a protein from left to right. That is the direction. So as it says here, we read the amino acid from the amino end. So if we had methionine, tyrosine, and serine, that is not going to be the same if we read it in the opposite direction as serine, tyrosine, and methionine. It's a different protein. So you must read from left to right. The other thing that we can say is that the methionine is the amino or the N terminus. So the N refers to this nitrogen, and the serine is the carboxyl or the C terminus, because this is the other end, so the carboxyl and the amino end. That is a key term that is used, again, constantly. Here's just another diagram showing you the same thing, but what is different? R groups are shown. That's the only difference. Here is central carbon, central carbon amino group, that was the carboxyl group, and there's the proton. This is the R group up here, and again, you can see the peptide bond that has occurred, and we've got them throughout. So going from the amino end, because this is that end here, N, amino residue, to the carboxyl down this end. This is the direction that we are talking about. The other thing that we need to talk about is that this polypeptide, so as we start to link more and more amino acids together, has a backbone. And so the backbone is that carbon, hydrogen, and nitrogen atom. But the R group forms the side chain because they protrude outwards from that central portion. What we also know is that most polypeptides are about 50 to 2,000 amino acids. There are smaller and there are certainly larger, but this is the general size. And when they are this size, this is what we would refer to as a protein. So typically we'll be talking about proteins because they're going to be this large. If they're less than 50 amino acids, they're called peptides. Proteins also have a mass, a molecular weight, and a unit for that is a Dalton. So 1 Dalton is 1 atomic mass unit. So if you've got a protein with a molecular weight of 50,000, that is 50,000 Daltons, or 50 kilodaltons, because remember, kilo equals 1,000. So we can just get rid of those 3 zeros and say 50,000 Daltons. And that's typically what we would do for something which is so big. Polypeptides can also be linear, but they can also be cross-linked. Chains can cross-link. And this is typically achieved through a disulfide bond. And remember, the only amino acid that had sulfur atoms that were of use in this particular configuration are cysteine residues. So if we look here, here is a cysteine molecule, or an amino acid, and here's another. We've got the sulfur hydryl group shown here, this sulfur hydryl group, and through oxidation or reduction, so remember, oxidation is loss, reduction is gain, so either of an electron or a proton. So if we want to lose the proton, we are losing it. This is oxidation. And so what we have is this occurring. Two sulfur molecules or atoms joining together, and we've lost the protons and electrons. If we want to break this to form this back, this is then adding or gaining a proton. That is reduction, and we can break it. So notice these arrows are showing the same length, which means that the forward and reverse reactions can both occur. We can either lose the proton or we can gain the proton. This is important within the cell because it allows for bonds to form and to break. Now, we're still talking about the primary structure, but remember, primary structure determines the secondary structure of a protein, and it is the secondary structure which determines the function. Now, when we look at the peptide bond, we find that it's essentially flat, and another way of saying that is planar. So all of the carbon atoms are on a flat plane. And the nature of the peptide bond means that it acts like a double bond. So you know that double bonds are rigid, and so what this does is that it prevents rotation around that central carbon atom. Remember, it doesn't affect the R group because the R group is outside of that peptide bond. So the R group can move, can twist. So, peptide bond is uncharged. Amino acids can also pack tightly, and we'll talk about this when we talk about RNA and DNA, which is quite different because they have a negative charge, so they can't pack together tightly. And so what this is showing you, as it says here, demonstrating the peptide bond is planar. You can see it lying on this flat plane. The R groups are shown in green spheres, and what this is meant to show you is that this is coming out of the page, and this is going into the page. So these are not lying on the same plane. The one on the left, the up, is pointing at you. The one on the right is pointing away from you. So it's not laying flat, which means that it's able to rotate. Okay. Now the planar peptide bond can also exist in two different configurations, a cis or a trans configuration. Trans is going to be the most common because you don't have steric hindrance. So what this means is that when two groups are on the same side, they're going to be closer to each other. If there's a charge-charge disparity, they're going to want to repel each other. That's not going to be stable. And so they will flip to the trans configuration. So you can see that here on the right, cis configuration, these two groups are very close. They're going to want to repel each other, and that's not going to be too useful for the cell. And so what we do is that this group is on one side, this other group is now quite far apart, and this allows for the two to be much more stable. Now, in contrast to the peptide bond, the alpha carbon amino group bond and the alpha carbon carbonyl group, these are pure single bonds. So this is where the rotation can occur and for the protein to fold in numerous configurations. Okay, I'm going to stop there because I want to move on to secondary structure and the remaining portions in the next recording. Okay, welcome back. So let's move on to the secondary structure. So polypeptide chains can fold into two forms of repeating or a periodic structure. The first one we've seen in the first lecture was called an alpha helix, and the other is called a beta pleated sheet. And these are connected through turns and loops. The secondary structure of a protein is therefore due to these alpha and beta helices and sheets. Okay. Now, this is an important point to note. In biochemistry, you'll notice that some terms are used often, but in different contexts. And this is one example. The alpha here is not referring to the central alpha carbon, it's just referring to the type of shape of that particular configuration. So don't get that confused. So the alpha helix is a rod-like structure, and you see it in this diagram below. Backbone, so the carbon, the hydrogen, and the nitrogen forms the center. The side chains protrude outwards. What are the side chains? These are your R groups. The structure is stabilized with hydrogen bonds, and you know that there's going to be lots of carbon, oxygen, and nitrogen and hydrogen groups, so lots of hydrogen bonding is going to be able to take place. The twist can either be right or left-handed, the helix. Right is the most common, and this is because you've got less steric hindrance, which is similar to what we spoke about previously with the cis and trans above. And the diagram below, this is an example of ferritin. This is a protein that's used for iron storage in cells, and you can see that 75% of this protein is an alpha helix. So this is an alpha helix, that's an alpha helix, that's an alpha helix, and that's the alpha helix. And the remainder, these other portions in the center, okay, here, is a turn, hook and loop, hook and turn, I should say. That just allows it to twist and bend. Okay, the other form is called a beta pleated sheet, and this is where the amino acids are pretty much fully extended. And what we have is that these are made by linking two beta strands connected by hydrogen or hydrogen bonds. And these strands can run against each other, so anti-parallel or in the same or parallel direction. So if we see here, these two are running opposite. We've got one going to the left, we've got one going to the right. And you can see the basic grouping with the shadows. These dotted bonds are hydrogen bonds. There's one there, up here, and so on. And here you can see that these two are running in the same direction, this way and this way. So this is anti-parallel, this is parallel. And again, you've got those hydrogen bonds that are keeping it together. Here's an example of a protein. This is a fatty acid binding protein. You can see that we've got one alpha helix, but it is mostly beta pleated sheets. And that's shown by these blue arrows. We've also got the turns and loops, just for allowing it to change direction. But you can see that the vast majority is a beta pleated sheet. Okay, what about the next level? So the next level for an individual protein is called a tertiary structure. So once a polypeptide has formed its alpha helix and beta pleated sheets, as we've seen above, the final shape is its tertiary structure. So in a linear protein, what you're going to see is that hydrophilic amino acids will dominate. So these are the ones who are going to like being in an aqueous environment. But in contrast, tightly bundled and compact globular proteins, so those that are compacted, what you'll find is that non-polar hydrophobic residues, like valine and methionine, are going to be on the inside of the protein because they don't want to be in an aqueous environment. And as a result, that hydrophobic nature will force them to be on the inside of the protein because they want to hide from the water. And similarly, polar and hydrophilic amino acids like aspartate and glutamate will want to be on the outside because, again, they want to be in the aqueous environment. So, again, that will force the protein to twist and bend until they are outside. This is the most energetically favorable option. And the example here is myoglobin. If you look at this diagram, you can see that you've got twists and folds. You can see this region, if I put an arrow there, this is outside. So this is going to be, let's try again, to the right. Hydrophilic, okay? And you can imagine that in here, if I put the arrow to the center, this is more hidden. And so this is going to be more of a hydrophobic region, okay? What you can also see, though, in this particular protein is that it has a heme group. It has what is called a prosthetic group. So it has a group that is bound to iron, and that is important for the functionality. If you took away this iron and heme group, this protein would not be able to carry oxygen as part of its function. So again, the diagram below tells you what's going on. Now you can see that this is for a single protein. There's only a single protein that is shown, and that's okay if we go to a tertiary structure. If we have more than one protein coming together, this is where a quaternary structure comes into play. So this is where several tertiary proteins come together to form a larger protein, and each one of these proteins is called a subunit. So if we look at hemoglobin, this consists of 2 alpha chains and 2 beta chains, and this is a tetramer because remember tetramer means 4, so 2 plus 2 is 4, so that's why it's a tetramer in its structure. But again, I just want to highlight these 2 terms, alpha and beta, not to be confused with an alpha helix or a beta pleated sheet. These are just the chains, they're called alpha and beta. And so if you look at this diagram, you can see that you've got 2 red and 2 yellow groupings. Now, don't know which is which, and it really doesn't matter, but if we say yellow is alpha and red is the beta chain, you can see that you've got these in this particular configuration. You can see the heme group and the oxygen group, one here, one here, one here, one here, and you've got them in this particular shape and configuration. So that's an important structure for that particular amino acid. Okay, let me stop there because we're near the end of the lecture, but I'm just noting this is getting a little bit long, so let's wait for the next recording to finish off this part. Okay, welcome back to probably the last recording for this lecture. So we know that amino acid sequence determines the final 3D structure. But as it says here, but how do we know this is true? A lot of our data is going to come from experimental studies. Scientists have made experiments to prove or disprove a hypothesis. So, in the presence of a reducing agent, so remember, disulfide bonds can either exist in the sulfurhydryl or the disulfide state, go backwards and forwards. These bonds can be broken, causing a protein to unravel. If we take away these reducing agents, the protein will reassemble back into a functional protein. And it does this in the absence of any external action. There's no other proteins, there's no other factors that are causing the protein to bend and fold back to the original state. So what does that tell you? It tells you that the sequence of the protein must determine the final shape of that protein. Otherwise, it will just remain in that scrambled configuration and not be able to do anything. So here is the example. Now, we're not using the protein that we were talking about here. Well, we didn't mention a protein, but we're talking about disulfide bonds. But here is the example of a protein called ribonuclease. So this works on ribonucleic acid. The native form means that it is undenatured. Okay, this is how it will be made by the cell. This is what we typically see. Now, look at the little spheres. One here, one here, one here, and one here. Each of these are disulfide bonds. So you see that you've got 4 disulfide bonds. And notice the numbers. They're the amino acids that are joined together. So they don't have to be next to each other. They can be quite distant from each other because the protein folds together in different configurations. Now, using different reagents, so urea and beta-mercaptoethanol, these are reducing agents. They will break the disulfide bonds. And we can see that they're broken because, remember, reduction is gain. The sulfur has gained a proton, and we've got this SH group, and it is unraveled, and so it is denatured. If we now say, here's the denatured protein, if we remove all of those molecules, so all of the urea and all of the beta-mercaptoethanol, we go from the scrambled version back to the original native configuration. How is that possible? It's possible because sequence determines shape. Okay? Those proteins, or those amino acids that want to be on the outside, are going to force the protein to configure to a shape that puts them on the outside. And similarly, those amino acids that want to be on the inside are going to force the protein to bend and twist so that they are on the inside of the protein. And that's what forces it into this most stable configuration. And when we have this shape, we have that functional activity. So it's a really beautiful system in the sense that the amino acids don't need any additional help. They're able to reform on their own. Okay, so that's the end of the lecture. Again, we've got the summary. Later on. Okay, so if we start off with now lecture 1. Now, one thing that I wanted to, before we start, is just remind everyone that when you go through the front of your notes, you'll see that study aims, and you'll notice that these individual aims are the key points that we're covering in the lecture topic, and these will also be the key points that we are focusing on tutorial questions. Be very honest with yourselves here. So where it says studied, this means you've got a good understanding, or if you have some questions and you need help, tick this box. Don't sort of think, oh, that'll be okay, I won't need that, without actually using it. And similarly, use this space here or anywhere within your notes to include those questions so that you can ask those questions. Remember, you don't have to verbalize them if you don't want to. You can put them into the chat. You can certainly just email them to me before the tutorial starts, and then I'll just read them out. I won't name a student. I'll just say, student has asked me this question. Here is the question that's been raised, and then we'll go through the content. I think it's really important. Highlight those questions that you've got. Act on them straight away. Don't let it prolong, because with biochemistry, the content's going to start coming fairly quick. It's fairly complicated in some aspects, and so if you don't understand the information, it will just build and build and build. Okay, so this first lecture was designed to give you very much a primer on a lot of the key points that we're covering and some of those basic things that we would expect you to know in a biochemistry context. So if I just go to stop doing that window, make sure I can see everyone. Okay, so we start off. What are the 4 classes of biological molecules and why are they considered essential for all organisms? So please feel free to unmute or put it into the chat function.
I have written proteins, carbohydrates, lipids, and nucleic acids.
Perfect, excellent, that's great, thank you. So they're all correct. I've got my answers here too, and I've just covered them up, but I'll take these off in a moment. And so why would you, if you understand that, why would you say that they're essential? So what would be the main, one of the functions of each of those biological molecules?
So I said proteins are important.
It's together that gives rise to the entire metabolic pathway. Carbohydrates, excellent source of energy. I've put glucose there because when we come to talk about metabolism, we will focus on glucose as the preferred energy or preferred carbohydrate, and it is the carbohydrate that is used within the cell for metabolism to generate energy. And again, when we are talking about energy analysis as a group, what do you think I mean by energy? Are we talking about a specific molecule when we refer to energy?
ATP.
Yeah, thanks, Anna. So yeah, ATP. Okay, so everything that metabolism, the whole point of metabolism is to generate ATP as the energy source. So I just opened up the chat function. So Allegra, Aaron, thank you for answering that as well. So lipids, energy reserves, absolutely. We'll talk about lipids. Lipids have a energy density which is about 2 to 3 times greater than carbohydrates. So you have glycogen. Glycogen is a carbohydrate store, but you have very limited amounts. It's not long term. Whereas lipids are a much more long term and much more energy dense store. And of course, lipids help to make the cell membrane. And when we come to talk later about prokaryotes and eukaryotes, we talk about internal organelles. They're also encased in lipids. So you've got lipids within the organelles, but also you've got lipids as the entire structure. And nucleic acids, so genetic material of the cell. This can be either RNA, it can be DNA. You've got a whole variety of those. And both of those can be used depending on what the nature of the organism is. But of course, when we're talking about ourselves, it's going to be DNA as the key source. Okay, so we're going to talk about all of these. We're going to talk about them in different contexts, but all of these will be covered throughout the biochemistry course. Okay, so if we move on, well, before I move on, are there any other questions? That you would like to raise. And I'll do this throughout. We'll go through a question, we'll answer it. If you've got anything else, please yell out. And if you've got a question, just put it into the chat. I've got that open in front of me. No? Okay. So if we move on to the next one, explain the significance of the 3-dimensional structure of proteins in biochemical reactions. How does changing the order of amino acids in a protein impact its function? So first of all, if we look at the first part, and I think this is an important thing to note, these questions are broken down into perhaps several parts. It's important for you to be able to identify each of those parts and address them individually. This will be very similar to the short answer questions that you'll get in assessments. So quiz, well, the quiz is going to be more multiple choice, but certainly in the exam where you've got these questions. And sometimes students, when they mark or answer these questions, will just read it and just think, oh, this is overall answer, and just make up the question in their own mind. So it's very important to highlight and link what's going on. So the first thing, significance of the 3-dimensional structure of proteins in biochemical reactions. Why do proteins have to have a specific... Well, the quiz is going to be more multiple choice, but certainly in the exam where you've got these questions. And sometimes students, when they mark or answer these questions, will just read it and just think, oh, this is... All of these, we're going to talk about them in different contexts, but all of these will be covered throughout the biochemistry course. Okay, so if we move on, well, before I move on, are there any other questions about this that you would like to raise? And I'll do this throughout. We'll go through a question, we'll answer it. If you've got anything else, please yell out. And if you've got a question, just put it into the chat. I've got that open in front of me. No? Okay. So if we move on to the next one, explain the significance of the 3-dimensional structure of proteins in biochemical reactions. How does changing the order of amino acids in a protein impact its function? So first of all, if we look at the first part, and I think this is an important thing to note, these questions are broken down into perhaps several parts. It's important for you to be able to identify each of those parts and address them individually. This will be very similar to the short answer questions that you get in assessments. So quiz, well, the quiz is going to be more multiple choice, but certainly in the exam where you've got these questions. And sometimes students, when they mark or answer these questions, will just read it and just think, oh, this is overall answer, and just make up the question in their own mind. So it's very important to highlight and link what's going on. So first thing, significance of the 3-dimensional structure of proteins in biochemical reactions. Why do proteins have to have a specific shape in order to function?
I wrote because the shape defines the function of the protein.
Yeah, that's good. Thanks, Hannah. Can you just extend it a little bit in terms of what is it about that shape? Okay, so proteins have 3-dimensional shape because they have to bind to a molecule. within a lock, when you have that match, it operates the lock. And the reason it works, and the reason that one key will open and another one won't, is because of that shape matching. That's the same thing with proteins. Proteins have specificity, and so they will typically bind to a substrate. Now, if there's another substrate which has a similar shape, it may be able to go in, and we'll talk about that later when we come to talk about inhibitors, but that's really the key point. Now, moving on to the next part, how does changing the order impact its function? So going to Kirsty's comment, changing the order of amino acids can change the function of the protein. Yes, and it can change the function because it changes the shape of that particular protein. Now, we'll be doing lecture 1's tutorial questions. Yes, so lecture 1 and lecture 2 today. Remember, the tutorial follows the previous week, so we're going through that content. Okay, so let's see what I wrote. So proteins have a defined shape, and that shape determines the shape of the active site. And remember, the active site is only about 5 to 10% of the entire protein. It's actually a really small amount of the whole protein. So the rest of it is really to help give you that conformation, give you that shape, so that the substrate is able to bind and come in. And so the active site is where the substrate binds and is converted into a product. Now notice the way that I've written it with square brackets. This is the convention that is used when we come to talk about it in a biochemical context. So biochemistry will say those square brackets for substrate and product, and you will see this time and time again, particularly in the enzyme kinetics lecture. Changing the amino acid sequence changes the 3D shape and hence the binding to a substrate. If the changes in sequence are minor, or they could be in a location which is distant to the active site, there may not be any consequence. So again, just think of a key. If you imagine a key, you've got the handle. If you make a change to the handle, it doesn't affect the active site of the key. You know, the ridges, they're not affected, so it makes no difference. So in some cases, those changes may have no effect. In other cases, those mutations might change the shape of the key. That is going to have a big effect. And it's the same thing with an active site. If you change the sequence and it affects the active site, the likelihood is that the substrate, A, no longer binds. If it does bind, it doesn't bind as effectively, and you need a much higher concentration. Or that something else binds. The incorrect substrate may now bind, and you get these off products, these side products, which is not what is expected within the cell. Okay, going back to you again as a group. Are there any... comments or any other questions, remember, feel free to unmute yourself or put it into the chat, or even if it's just a thumbs up in the screen if it's all good, or if you've got a question. Thanks, Chip Allegra. Okay, let's keep going. So next one, how do lipids contribute to the formation of the cell membrane? So Zoom user, hydrophobic and hydrophilic regions forming the inside and the outside of the cell. Correct. So they've got those different domains, and because we are an aqueous, or cells exist in an aqueous environment, so a water environment, those hydrophobic and hydrophilic regions are going to constrain and force it to orient within a particular shape. And that's what leads to this formation. So first of all, lipids pack very tightly because of this hydrophobic and hydrophilic association. When they're hydrophobic, this is the bit that wants to be away from water. So if you imagine a sphere, and you've got the lipids, so a ball, you've got the lipids on the outside, those hydrophobic regions are going to be on the inside of the cell, all right, or the inside of the membrane. And if you put those two regions against each other, they're going to hide against the water. The hydrophilic are going to be on the outside. So you've got the outside, which is to the outside of the cell, but you've also got the inside of the cell. But because you've got a double membrane, you've got that space in the middle where the water is excluded, and then you've got the water on the inside, so it allows you to have that separation. And so that's what that last part is. It's talking about the internal and the external location of a cell and an organelle. So remember, cells have this structure, but within eukaryotic cells, we have organelles. They also have this lipid membrane, and this helps to contain enzymes, proteins, etc., and to allow those functionalities to occur within a specific location. Okay, any questions? Okay, next one. Outline the flow of information in the central dogma of biochemistry. What do you think? So where do we start? is it nucleic acids, is it proteins, is it lipids? What's the order that we talk about? So DNA, correct? Transcription, translation, Joe, DNA to RNA to proteins, perfect. So you start off with your nucleic acid. Our nucleic acid is DNA, so we take that DNA is then transcribed into RNA. That RNA can take multiple forms, and Hannah has written DNA to messenger RNA, which is correct. That messenger RNA is a form of RNA that allows the cell to read it and in turn generate a product, and that product is going to be a protein, because the protein is what does the function. So DNA encodes for RNA, and I've just written RNA, mindful that there are multiple types. We will talk about ribosomal RNA, messenger RNA, and transfer RNA later in the semester. They're all involved in different aspects, and then that RNA is used to encode a protein. One thing that I want you to think about, and we think about the changes in amino acids earlier, change in DNA can hence change the RNA sequence, and that in turn can affect proteins, because we're going in that order. That ability to affect a protein can also then be linked to cancer, so cancerous states where you have a mutation. So the reason we talk about mutations in a negative context, if you imagine ultraviolet rays, okay, next few days is going to be hot, if you go to the beach, you need suntan lotion. Why? Because you want to protect yourselves from the ultraviolet rays, because that UV light can damage your DNA. That damage can then affect the message that is being made and ultimately the protein. And you may generate a protein that is abnormal, and that leads to a pro-cancerous state. Now, Erin, DNA replicates, transcribes into messenger molecules, translates into a protein which perform biochemical reactions within the cell. Perfect. That's really nice. That's a nice way of explaining it. Well done. Thank you. Okay. Any other questions for this one? Okay. I think, no, not more. This one, distinguish between prokaryotic and eukaryotic. These are just those big ideas. What you'll see with the biochemistry content is with the more complex processes like protein synthesis or replication, we start off with prokaryotes, give you the foundation, and then we apply the same approach to the eukaryotic setting. What you will notice is that the prokaryotic system is probably a little bit simpler, but the basic process in eukaryotes is the same. There are just more players. There are more proteins, there are more factors that are involved, but the basic idea is the same. So my suggestion from a studying and understanding perspective, make sure you understand what's happening at the prokaryotic level first, and then when you move over to the eukaryotic, you'll see, oh, it's very similar. The only difference is, oh, here's more proteins, or here's a different start site, or here's a different amino acid that is part of the protein. And as soon as you do that, rather than having to remember double, you're just remembering those minor differences, and then you can really combine them together. I think it's going to make it much more efficient for you and easier to move between the two sections. So back to you. What are some of the differences between prokaryotic and eukaryotic cells? So Kirsty, eukaryotes are membrane-bound organelles. Thank you. Thanks, Haley, for the same comment. Eukaryotes membrane-bound. Yep. What else? Prokaryotes lack organelles. Okay, so Erin. Prokaryotes membrane-bound organelles, single circular chromosomes found in the cytoplasm, cell wall enclosed net. Eukaryotes membrane-bound organelles, linear DNA found in the nucleus, and they have a cell membrane. Perfect. And Ira, eukaryotes can reproduce sexual meiosis or asexual mitosis. Yeah, that's good. Probably not like anything, but is correct, so that's well done. So, I think we've covered most of the points there. So, prokaryotes lack organelles. So, I've got this later. The reactions in turn can occur in all locations. What you'll see within eukaryotic cells is that by having organelles, you can segregate functionality. You can segregate the function of energy formation of ATP into the mitochondria. Whereas with prokaryotes, it's going to be more distributed. There are some advantages, there are some disadvantages to those approaches. So one of the advantages of a prokaryotic cell, it doesn't need to move things around. Things can occur very quickly. Whereas eukaryotes, you make something in one location, it has to be transported to another location. That all adds a delay. And so you know that prokaryotic cells will divide very quickly, relatively in comparison to, say, a eukaryotic cell. Each one of those additional layers delays and adds time. Membrane-bound organelles within eukaryotes, and reactions are associated with those organelles. So that's an important one, in addition to the comments that we've got in the notes as well. So that's really good. Thanks very much for interacting. And the last one is explain the key functions of mitochondria: smooth and rough endoplasmic reticulum, Golgi, endosome, and the lysosomes. So let's start with the mitochondria. I've already hinted as to what that is in my answer to the previous question.
Generate ATP.
Generates ATP in the cell. Thanks, Aaron. Perfect. This is the powerhouse. Okay, this is where ATP occurs. This is how it would go through. So, if question 5 is an exam or test, how many marks will it usually have? Question 5, that one is probably only 2 or 3 marks. It's a fairly short and simple question, but you probably wouldn't get something like that. I'd probably go a bit more in depth as opposed to something like that. So Zoom user, mitochondria, good energy source of energy, ATP production. Okay, perfect, good. Now, ER, we've got the smooth and the rough. So what do you think in general is the function of the ER? And then the next question is, well, what is the difference between smooth and rough? What else is being attached in that context? Chemical modification, perfect. So modification of proteins. And Joe, rough in the place of reticulum, contains ribosomes for protein synthesis, smooth performs chemical modification, perfect. Well done. Okay, rough has ribosomes for protein synthesis, great. So the ribosome is what does the protein synthesis, but what you see is that we said proteins are modified, and some of that modification can be, it's called post-translational modification, so after it's made, things are added, groups might be added. So you can actually have carbohydrates added to a protein, you can have lipids added to a protein. You can have specific groups added to proteins. The point of that is that by adding those different functional groups to a protein, it acts as a marker, and it tells the cell, this protein, if it's a lipid, maybe it has to go to the cell membrane. If it's a carbohydrate, maybe that's part of its functionality. It's required because it has to bind to an enzyme that facilitates the process. Okay? Adding a phosphate group and taking it off is another example of modification of proteins, but that's one that occurs very actively within the cell, and it's used as a control mechanism, not necessarily within the endoplasmic reticulum. But we'll talk about that a lot. Now the Golgi, this is also a spot where those additional changes can occur. So you've got that further processing, so as I mentioned, the carbohydrates. Now we've got the endosome and the lysosome. What do you think the endosome is doing? The name is actually linked to one of its functions, so that is an easy way to remember what's actually happening. Entry to the cell, yeah, a bit more detail, what does that mean? So if I say the term endocytosis, what does that mean to you? So if you think about phagocytosis, endocytosis, taking material into the cell, Hannah, correct. So external, so Erin, perfect, material from the outside of the cell moves to the inside of the cell through endocytosis. Thank you again, so Ira, thank you, take material from the external environment, puts it back into the cell, correct. This is a way that the cells are able to take something which is outside, internalize it, and use it, assess what's going on, for example, and bring it to the inside of the cell. So Haley, take useful materials like vitamins. Yeah, vitamins probably more transported in, there might be binding, but yeah, if they're associated with other proteins, the other thing that I'm thinking of would be, say, cholesterol. Okay, that's a good biochemical example where you would have internalization. And then the last one is the lysosome. When you take something in, you use it. What do you do with how do you get rid of it? What might be a way of getting rid of it? So lysosome digests waste, recycled materials, the stomach of the cell. Thanks, Joe, that's a good way of putting it. Degradation, meiosis, breakdown components. Not sure about the link with meiosis. Don't think that's correct. So I wouldn't go down in terms of the reproduction, the cell division side of things. I think Joe has hit it more accurately. That lysosomes digest internal molecules for destruction and removal. This is also a way of actually destroying organisms. So maybe Zoom user, that's what you're referring to, breaking down of components. Certainly from an immune perspective, cells of the immune system can ingest bacteria, and in doing so, they can degrade them and get rid of them. So yes, okay, perfect, good, you're on the right track. And Erin contain digestive enzymes used to degrade ingested material, perfect. So that's important. And Emily, for the endosome, if you've got a question about the test exam, do you need to mention the secretory granule? And the question should be typically quite specific, so it'll tell you whether you need to go into more detail or not. But yeah, that's Charlotte put the smooth down process breaks the rough out proteins, does the smooth. I think you can have transport of proteins into the smooth. The rough is where it's direct. I'm not sure if it's also occurring. It might also be drugs. I do need to perhaps double check that one. But I do think you still have some proteins within that space. There's probably a bit of overlap between the two, but that's a really good question, very specific question. Okay. Any other questions from the group? Feel free to, as I said, feel free to unmute yourselves if you're happy to, or anything through the chat function, or just let me know through some sort of reaction on the screen. Chat, thank you. Thanks, Ellie. Thanks, Zoom user, Erin. Okay, so Sheena, thank you. So if there's no other questions for this one, what I'll do is I'll stop and we'll move to the second lecture. So the second one was amino acids and proteins. So again, you've got those linkages there. Notice we talk about structure, it's in the question. The 1 and 3 letter codes, we've got that in the tutorial. Levels of organisation is also in the tutorial. The one that's not is the process of denaturation, but we will talk about that later. So again, it's just a highlight to you. You should see similarity between the questions or the study aims, and then I align that with the tutorial questions at the end. And also notice, the questions that we are asking, these are big picture questions. I'm not asking you for very minute detail or things that are not so critical. I have tried to keep it on the big picture, the big idea. And similarly, any exam question is going to be on a big question idea. It's unlikely to be on a very small, specific topic. We're not here to trick you, we're not here to confuse you. It's all about, do you understand the big picture? And that's actually one of the best questions you can ask yourself after every lecture, is do I understand what this entire lecture is about? What are the key features of this lecture? Could I explain those key features to your parent, whether it's charged or uncharged, on the nature of the pH? So again, here, just highlighting, some of these diagrams are really useful. So this shows it with you, stays the same. In the notes, I had the same thing, but it was just oriented the other way. It was 180 degrees. And then the vinarine and lysine, because when you start to see this, particularly as we progress further and further, you're going to need it. And I do know that when you start to do some molecular biology courses, so if you've ever taught with Ben Roberts, he will expect you to know this and to have them so that you can get tips. Okay, so describe the primary sequence of a protein and why it is important. So first of all, what is the primary sequence? When we say a protein has a primary sequence, what are we referring to?
The specific sequence.
Joe, amino acid sequence, perfect. That's the most basic idea that we can start off with, is just what is that linear sequence of amino acids? Thanks, Emily. Linear is a good word to add. So it's the sequence, and it is the linear sequence. We're not worried about any conformation or anything. It's just like a bit of spaghetti. Just got amino acids and they're just sort of flopping around everywhere. So sequence of amino acids linked together via peptide bonds. Thanks, Haley. And Zoom user, order of amino acids in a protein determines the subsequent 3D shape. Fantastic. You're all spot on. The primary sequence is what you'll see in the textbook, right? Someone will write something in and it'll just be a string of letters as a paragraph. And you look at that and go, well, that's really informative, but actually it is. It's telling you this is the actual sequence of the protein. It doesn't tell you anything about the shape, although we know the individual characteristics of the amino acids, and there are programs that you can put it into that will draw and constrain the protein. Now, there is an AI bot. I forgot what it's called, not Gen AI. There's, I've forgotten it now, but there's a company in the UK that can take the sequence and the shape of a protein. And we said that if we change the DNA, we change the message. We change the message, we change the protein. Okay, secondary structure. So what is the secondary structure and why is it important? They're twisted and they're more packed together a bit, chains protrude outward. So the backbone is that core portion, the side chains, we're referring to the R groups here. And so the nature of the R group is going to be something which is hydrophobic. Hydrogen bonding is very, very important. Now, hydrogen bonds individually are actually very weak, and that's by design. Hydrogen bond is a weak interaction, but what that means is that a weak interaction, you don't need a lot of energy, either in the form of ATP or even just heat in the body to make and break bonds. And that's important because you want, from a biochemical perspective, you want to be able to make and break bonds so reactions can occur. But individual bonds that are weak, if you have lots of them, they all combine. So if you've got a bond that's only 1% of energy, that 1%, but you've got 100 bonds, then it all becomes very, very strong. And so that's the advantage by having these multiple bonds and allows for different areas to open together. And clearly you've got carbohydrates, or not carbohydrates, carboxyl group, and an amine group, oxygen and hydrogen and nitrogen atoms, able to make these hydrogen bonds very effectively. Okay, tertiary structure of a protein. Why is it important? Final 3D shape of a protein allows something to occur. Okay, perfect, thank you. So it's that final shape. And the key point in that answer as well, single protein. So Haley's structure results from final folding of protein through interaction between R groups, correct. So you have those different groups. Remember, these are not static, they're moving. You have those groups, they come together, and they come together when they're at the lowest energy state. They are at their most stable configuration. Stability is going to be affected by that hydrophobic and hydrophilic interactions. If you have a twist, so that's part of the protein folding that is taking place. So final course of a single polypeptide, that's the other key word to put here. I haven't got a question on quaternary, but I will touch on it in a moment. We're talking about a single protein in this shape. Now, remember, we have linear proteins with mostly hydrophilic amino acids, and those proteins that are very tightly bundled, this is where you start to have that segregation of hydrophilic and hydrophobic regions coming together. Now, I just want to quickly zoom back to this diagram. This was hemoglobin. This is then the example of the quaternary structure, and I've got it here as well. You've got multiple groups. So if we look here, we've got a single protein. So this is the tertiary structure. Here we've got multiple proteins, so 4 proteins that have come together. So 4 tertiary structures have come together to give rise to a quaternary structure. What this allows you to do is to have a larger protein complex. That larger protein complex maybe has a better functionality or requires this 4 functionality to occur so that the substrates come in, or by having 4 molecules, you've got a greater capacity, in this case for the hemoglobin, more oxygen carrying capacity in this molecule. We will talk about proteins existing across tertiary as well as quaternary structures. So we'll have single proteins. When we come to talk about replication of more advanced complexes, you'll start to see proteins coming together and will be in this quaternary structure. So lots of individual proteins coming together and perhaps forming complexes. When you have a complex like this in a quaternary state, any defect that occurs in one subunit, so if we imagine that this red unit here had a mutation, that could be sufficient to break all of this apart and therefore to make it unstable. So are the answers to structure importance the same from primary to tertiary? I'm not quite sure I understand your question. Structure is going to be important for all proteins because the structure is what determines its ability to interact with substrate. Is that what you meant? Good, thank you. So that's really the importance of that. So it's really just to talk about the function of these, and certainly later on when we come to talk about signal transduction, we will talk about 2 proteins or 3 proteins that have to come together for a signal to actually be transmitted throughout the cell. And the reason I'm putting my fingers sort of going backwards and forwards is that when you have 2 receptors, for example, they might be far apart and they're not binding to their ligand. When the ligand binds to one, it causes the other one to come together, and then you have a dimer, or you might have 3, so you have a trimer or a tetramer or whatever. You have multiple complexes coming together. And again, another example of quaternary. It's practical, but ask questions, engage. Please do not ignore this advice. Engage with the content. Okay? Ask for help, get that help early,